Well, I did my PhD in biomedical science at Auburn. I had spent some time at University of Alabama, Birmingham, working on the genetics of cystic fibrosis. But then I had an opportunity to come to Auburn and work on this feline model of a rare inherited disease in kids, and it actually occurs in cats too. And I was so fascinated by that, by the possibility of learning something in the animals that could be translated into human medicine, that I just wanted to work on that, and that's where I did my PhD. So after that, I spent some time in different places, mostly UAB, but then came back to Auburn and did a postdoctoral fellowship, and have just kind of been in this track for several years, moving from assistant professor to associate professor to full professor. And then the opportunity came up to apply for the position as director of Scott-Ritchey, and so I put in my application and fortunately was chosen for the job. So I've been in place since June of 2020. Scott-Ritchey is almost unique in veterinary colleges across the country because it's a research facility that is affiliated with the College of Veterinary Medicine, and it's privately endowed. So there aren't very many of these across the country, and our models of diseases of animals and humans are quite unique. But the way the whole center came to be is that there were two people who were really interested in doing research that would benefit companion animals. One of those was a guy named Kenneth Scott, who was a wealthy shipping guy. He had a lot of money, but he loved doing hunting trials, field dog trials, locally in Alabama. So he started a gift matching program where if someone gave a gift to support companion animal research, he would match it. So that's where the Scott comes from. And then Eleanor Ritchey is the second part of the Scott-Ritchey name, and she was the sole heiress to the Quaker State oil fortune. She loved dogs, and she went around the streets of Daytona Beach, Florida, and picked up stray dogs, ended up with about 150. And so she wasn't married, didn't have kids, had some distant relatives, but she left her entire fortune in her will to Auburn University to start what eventually became the Scott-Ritchey Research Center. The center was the brainchild of a neurologist here named Dr. Horline. Dr. Horline is legendary at Auburn and has students who have gone on to greatness all over the country. But his vision was to have a companion animal health research center. His interest was neurology, and so the early focus of the center was on neurology. That has remained over the 35 years of the center, but it's branched out. We've branched out a little bit more since then. The person whose vision this whole center was named Dr. Horline. And he was a neurologist that is really legendary at the College of Veterinary Medicine. He's educated veterinarians that are now all across the country doing great things. He was a neurologist, as I said, and so his primary interest was neurological diseases. For example, dachshunds that have spinal cord injuries. He was big on treating those. So Dr. Horline had this vision to create this research center, and so our first focus of the center was neurologic disease. That's carried on through the decades. The center's been around for 35 or 40 years, and neurological disease continues to be one of the main focal points of the center. Those neuromuscular diseases now are being treated by a type of gene therapy that's based on a naturally occurring virus. The thing is, though, that the majority of that virus, 95% of it, has been engineered out so that any genes that were in the virus that would make it harmful are gone, and we're using the virus just as a shell to deliver gene therapy. We've taken all the good parts of the virus and made them, put them towards our use. So that's what we're doing today, is using engineered viruses to deliver genes to treat different kinds of diseases. Neuromuscular diseases are one of those. So Tay-Sachs disease was really first discovered in 1881 by two neurologists who saw these kids who would be born normally and develop normally for a few months, and then they would start to miss developmental milestones, and then they would plateau in their development, and then they would start to regress. And it would be a slow, well, not necessarily slow, pretty rapid regression to death at about 3 years of age. So that's been 130 years ago that we've known about, actually 140 now, that we've known about Tay-Sachs disease. There's still no effective treatment for these kids. They invariably are born normally, they develop normally for a few months, they plateau, they get worse, they spend most of their lives in a reclining stroller because they can't swallow well, and the reclining stroller helps them swallow a little bit better, but eventually the kids have to have stomach tubes because their swallowing is so compromised that they end up aspirating food, they end up with pneumonia, and pneumonia kills the majority of the kids. With the stomach tubes, you can avoid pneumonia, but the end result is still the same. We knew in the mid-1900s that this was an enzyme deficiency. We discovered that the cause of the disease was the lack of a certain enzyme. And so the next logical thing to do to try to help the kids was to replace the enzyme, just purify the enzyme from animal tissues or something like that, and then infuse it. Well, the blood-brain barrier prevents that from doing anything because the blood-brain barrier filters out the purified enzyme, as it does for 95% of the rest of what's in your blood. It's a protective mechanism, and it's good. If there are toxins in your blood, you don't want those in the brain, so it's good in many cases, but in this case, it prevents us from being able to treat the kids. So other ideas came along. Well, maybe we do organ transplantation, like liver transplantation, and we use a normal liver that can provide cells that will migrate up to the brain and deliver. No, that didn't work. Bone marrow transplantation, using these purified synthetic particles called liposomes to deliver enzyme to the brain, that didn't work. And it's been a series of over and over again having an idea, testing it, and finding out it doesn't work, and then moving on to something else. This has occurred decade after decade after decade. One of the big problems, as I said, is getting into the brain. And so in the 2000s, we found out that this particular type of gene therapy injected intravenously, IV, can get into the brain. And so we thought, well, this is the next thing we should try. We began treating animals with this gene therapy vector that's based on a virus that injected IV can get across the blood-brain barrier, and it started working really well. So in most untreated cats with Tay-Sachs disease, they live about 4.5 months. In the gene therapy treated cats, they're living about 20 months, so 4-fold increase in lifespan. The big thing for us is the improvement in the quality of life. So untreated animals develop these whole body tremors, and their whole body is tremoring back and forth, and they can't stabilize themselves well enough to eat very well. They can't do things that normal cats do, use the litter box. So they're pretty much, for the last month of their life or so, they are totally dependent on the people who are caring for them. So instead of that disease course in untreated animals, the treated animals develop no whole body tremors. What they end up dying of is that they lose function in their hind limbs, and so they can't stand any longer. But they're very comparable to a paraplegic, somebody in a wheelchair. And so they live a really pretty good quality of life, and they live 4 times longer than untreated, which was dramatically better than I ever expected. I thought when we started the gene therapy studies, maybe we'll be able to double the lifespan. If that would be like a grand slam home run if we could double the lifespan. Well, we've ended up quadrupling the lifespan and dramatically improving quality of life too. So that has been a big step forward in the development of therapies. There's a similar disease called GM1 gangliosidosis It's not as well known, so I don't usually start off talking about it, but it's very similar to Tay-Sachs disease. It would be like a close cousin of Tay-Sachs disease. And those animals live to be about 8 months of age. Well, we've treated them with gene therapy as well, have 2 cats that are still alive at 11 and 12 years old after treatment. The average increase in lifespan is 8 times, and their quality of life is dramatically better too. That work has led to, along with some studies in mice with our collaborators, has led to a clinical trial at the National Institutes of Health. So 12 kids have been treated with gene therapy that was based on what we tested in mice and cats, and all but one of those kids is showing clinical improvement now. So the next step that we hope for that work is FDA approval of the gene therapy, and there are still a couple of steps and hurdles that have to be met for that, but we're well on track to make that happen. Henry Baker was my mentor for my PhD, and he and his wife found these cats. They discovered these cats in the Southeast years and years ago, like early 70s. And so they maintained these animals, and they did a lot of those earlier studies that I was talking about with purified enzyme treatment and bone marrow transplantation. They did a lot of those studies. It's a naturally occurring disease in the animal population, and so I still get calls every now and then from veterinarians in the Southeast that want us to test for one of these diseases, and it's still out there. The advantage to the cats is that they give us a much truer picture of the disease in humans than something like a mouse. For example, the mouse model of Tay-Sachs disease doesn't even show clinical signs, because the absence of the enzyme in mice, for a lot of complex biochemistry that we don't even want to get into, it doesn't affect them in the same way that it affects people. So a mouse with Tay-Sachs disease will live a normal lifespan, but cats with Tay-Sachs disease have these severe clinical signs, and so they're much better model. The other thing is the mouse brain is about 1,000 times smaller than the brain of a human child, whereas the cat brain is only about 50 times smaller. So it's a whole scale difference between going from mice to humans and going from cats to humans. The scale up is much better modeled in the cat than it is in humans. Also, the mouse brain is very smooth. If we looked at a human brain and it looked like the mouse brain, we would say this human has a severe developmental defect. Mice just don't model human disease very well, and so the cats are a much better model. When we first started thinking about the project, my first hesitation was that we'd been working with these rare diseases for so long, and it was really quite hard to get others interested in helping us treat the rare diseases because they're rare. And so my first thought was nobody's going to be interested in rabies because there are only 4 or 5 people a year in the United States that die of rabies. Well, that's true, but around the world there are 50 to 60,000 people per year that die from rabies. Places like China, India, South Africa still have a dramatic problem with rabies. We had a visitor recently from Egypt, and her parents' neighbor back in Egypt recently lost a child from rabies because they don't vaccinate their animals, and so kids are going to go play with dogs, and dogs are eventually going to bite them, and it happens over and over again, thousands and thousands of times a year. So we started looking at ways to treat rabies once it has progressed to the point of being an encephalitis, something that is clinically obvious. Most people think that once rabies has developed clinical signs, it's over. There's nothing you can do, and that's pretty much true. There are a couple of extraordinary measures that have been implemented, like putting people in induced comas and hoping that they'll be able to make it through and survive, but it's really, once somebody develops signs of rabies, they will die within 10 days. So we started looking at ways to use the gene therapy platform that we had developed for Tay-Sachs disease to also treat rabies, because we knew that it gets into the brain, and that's one of the big hurdles for rabies treatment, just like it is for Tay-Sachs disease. We started expressing an antibody from the gene therapy vector that we knew would neutralize the rabies virus if we could just get it into the brain. And so those studies have progressed through mice and into cats now, and we're getting ready to start what's called a challenge study with a collaborator in New York to demonstrate that it really does treat the disease once the disease has developed. So I'm really excited about that. I don't think it will have as dramatic impact on people in the United States, but in other places around the world it'll be a dramatic improvement. At Scott-Ritchey, we're trying to use the same basic gene therapy platform that has been used to treat other diseases to also treat cancer. And the leader in those studies at the center is Dr. Bruce Smith. Bruce is the director of the Auburn University Research Initiative in Cancer, which brings together cancer researchers from all across the university, not just the college. Bruce's particular research in his lab is using the common cold virus. It's an adenovirus that's been highly engineered, and so that it no longer causes the cold, but it's used to transmit genes that can treat cancer. One of Bruce's focal cancers, or one of the cancers that he's focused on, is osteosarcoma. This is one of the things that I'm really excited about for Scott-Ritchey going forward, is taking something that we have worked on in the lab and translating it into animal medicine. So Bruce has done a clinical trial in dogs with osteosarcoma, which is a bone cancer, really bad cancer. The dogs generally have to have their limb amputated, and most of them die within a year after amputation, no matter what is done. Chemotherapy, radiation, doesn't matter. It's a really aggressive disease. So Bruce took a gene therapy and did a clinical trial at the Small Animal Teaching Hospital, just like you would do in human clinical trials at a human hospital. So I'm really excited about that type of thing going forward. He's also looking at lymphoma and other types of cancer. We have another very talented researcher, Payal Agarwal, who's been in Bruce's lab for many years. She's developing all kinds of new cancer treatments. One of those is called CAR T cells, so isolates T lymphocytes from animals with cancer and then treats them with gene therapy outside of the body and puts them back in. A lot of these CAR T therapies have been FDA approved for human cancers, and so I'm really excited that maybe the same thing will happen for animal cancers as well through Payal's work. Yeah, I think the center really should be focused on making an impact in animal health, and this is one area that we can make a huge impact in, because obesity is a problem across dogs and cats. Everybody who has had a cat at home will have had a fat cat. They love to just sit around and eat and do little else, especially if they're indoor cats. I've had several of those at home myself. Dr. Emily Graff is an adjunct professor she's in the Department of Pathobiology, but she's an adjunct professor at Scott-Ritchey, and she is an expert in feline obesity, along with Dr. Robert Judd, who is a director of the Boschel Diabetes Program. So they are really first-class experts on obesity. Emily has a colony, or has had a colony, of cats that we just called the fat cat colony, because they sat around and ate all day and became fat. There are differences in cat obesity versus human obesity, and Emily is exploring the differences to try to find ways to treat both. In other words, why do humans have an inflammatory component of obesity that cats don't, and vice versa? Another thing that she's doing is looking at different ways to treat obesity, so looking at it as the disease that it is. She's looking at different methods of treating obesity and getting that problem under control before it develops into something more serious like diabetes. I'm not a veterinarian, but I'm proud to be associated with veterinarians because they have known about One Health for a long, long time. One Health has become sort of a catchphrase in medicine recently. Finally, people have started to realize that animals and humans have basically the same diseases, and what you learn from one can be applied to another. Veterinarians have known that for literally decades. They have recognized the commonalities. So Scott-Ritchey really could be considered a One Health Research Center because most of what we study are diseases that occur in both animals, companion animals, and humans. Good example is the Tay-Sachs disease. It occurs in cats infrequently. It occurs in humans rarely, but what we've learned in the cat studies has been applied to the human disease. Not only have we learned how to treat Tay-Sachs disease in cats, which, as I said, is rare, we've learned through those studies how to treat brain diseases in cats in general, because we know how we can deliver genes to the cat brain now. We know that from the last 15 years of studies that we've been doing at Scott-Ritchey. So if another condition, brain condition, comes up in cats that we want to treat, such as brain cancer, for example, we know how to get genes into the cat brain because of all the studies that we've done. Same kind of thing with dogs, that we know how to deliver genes to dogs now because of these types of studies that have been done in dogs with cancer and other things like that. So one of the big issues in treating any disease is delivery. The delivery platform is critical. As I said with Tay-Sachs disease, we knew in the mid-1900s what the cause of Tay-Sachs disease was. It was an enzyme deficiency. We just couldn't deliver anything to treat the disease until the gene therapy platform came along. So I think what we're studying at Scott-Ritchey will impact certain diseases and have big impact, hopefully, but we're also studying a delivery platform that others can take and apply to lots of other different kinds of diseases. When I first thought about becoming director at Scott-Ritchey, I went back and looked at some of the other people's visions for Scott-Ritchey. One of those was Dr. Horline, and I read some of the early things that he had written from Scott-Ritchey newsletters of many, many years ago. And really, my vision for the center hasn't changed that much from what he thought all those years ago. He even mentioned inherited neurologic conditions like Tay-Sachs disease in one of his newsletters. And this was long before these cats ever came to Scott-Ritchey. So it's fascinating. Dr. Horline's vision and other directors' visions and my vision is to take our research and make an impact in animal health and human health. A lot of people do a lot of really cool, innovative studies on the benchtop or in cell culture that never or decades later make an impact on human health or animal health. I want us to be more translational than that. Translational is the word that we mean to when we say we want to take research from the benchtop and make an impact in actual living beings. And that's what we want to do going forward. I mentioned Dr. Smith's clinical trial in osteosarcoma. That's what we want to do going forward. We want to take promising treatments that we've developed in the labs at Scott-Ritchey and move those into actually treating animals and people with terrible diseases. If we're not making an impact on health, then what are we doing? It's fun to go into the lab and create some new innovative thing that nobody's ever thought of before. But if you're not making an impact, really, what's the point in doing that?